So this lecture segment is about the structure of DNA. And you'll recall that DNA is composed of nucleotides. Nucleotides consist of a pentose sugar, either ribose or deoxyribose, and a phosphate group and a nitrogenous base. So looking at the pentose sugar, you can have either ribose, shown here on the left, or deoxyribose, shown on the right. The proper name for deoxyribose is 2'-deoxyribose. Nobody ever calls it that, but that's the full name, because on the 2' carbon you're missing the oxygen that would normally be attached via the hydroxyl group. So at the 2' carbon you're missing an oxygen. The phosphate group will be attached to the 5' carbon through an ester bond, and the nitrogenous base is attached to the 1' carbon of the pentose sugar, and the nitrogenous base is either going to be categorized as a purine base or a pyrimidine base. And the purine base has the 2 fused ring structure, shown here, and that includes either adenine or guanine. And the primary difference between adenine and guanine is mostly in the functional groups attached. For example, on this number 6 carbon here, on adenine you have an amino group. On guanine you have an amide group, actually, but you have the double bond O. And in the pyrimidine structure you just have a single ring structure, a 6-membered ring. Cytosine, thymine, and uracil are classified as pyrimidines. Again, the difference is mainly in the functional groups attached to the ring structure. Now, nucleotides that are found in DNA, and for that matter RNA, actually have pretty complicated names. So a nucleotide that contains adenine is called deoxyadenosine 5-monophosphate, but we usually just call it A, or we sometimes just call it adenine, but what we really mean is a nucleotide containing adenine, or deoxyguanosine 5-monophosphate, or deoxythymidine, or deoxycytidine. But these are the full names, and we just usually refer to the whole nucleotide by the name of whatever base it has, so A, C, T, G. But you don't need to memorize the names, but these are really the full names of these building blocks. So when you look at a polynucleotide strand of DNA, typically you're going to have actually 2 strands. That's why it's called the double helix. You'll have a strand running in this direction, and the the nucleotides are attached by phosphodiester bonds. And then you have another strand running in the opposite direction, and those nucleotides are attached to each other by phosphodiester bonds. But the strands are attached across to each other by hydrogen bonds, which are shown with the ... right here. And so guanine and cytosine will make 3 hydrogen bonds with each other. Adenine and thymine will make 2 hydrogen bonds. And so all the way up the double-stranded DNA, you'll have the hydrogen bonds holding together the 2 strands. For RNA, which is typically single-stranded, you have the phosphodiester bonds that hold the sugar-phosphate backbone together. And then the nitrogenous bases are sticking out to the side. Of course, RNA has the uracil instead of where DNA would have thymine. So the double strand, or the double helix of DNA, has certain measurements spatially in what's considered the tertiary structure. When the DNA coils around, makes the helical shape, you have measurements like this. Okay, you have 3.4 nanometers per turn of the helix. You have 0.34 nanometers is called the rise, and the whole diameter is 2 nanometers. On one end of the molecule you have the 5' end. It's really the free 5' phosphate end and the free 3' hydroxyl end. So on the end that has the free 5' phosphate, that's called the 5' end, and the end that has the 3' hydroxyl that's free, that's called the 3' end. Okay. DNA has different forms. The B form is the traditional Watson-Crick structure that was described by them in 1953. It's considered the biologically relevant form of DNA in the cell. The A form DNA, which exists under dehydration conditions, will form, it's a right-handed helix. It's more compact. It has different measurements from the B form. And the Z form DNA is a left-handed helix that was first observed when synthetic DNA was created that contained only the cytosine and guanine base pairs, and it naturally will turn in a left-handed direction. Left-handed means counterclockwise as you look down, as the helix spirals away from you, there's a counterclockwise turn. That's what left-handed means. It's not clear whether there is Z-form DNA in the cell, but if there is, it could be very interesting in terms of what that might do to, for example, gene expression or control of gene expression. And here's some pictures. You don't need to memorize the measurements of this. Remember that the B form DNA has the measurements I showed you earlier. The A form is more compact, sort of squished down from the top maybe, and the Z form, you can see it spirals in the opposite direction. So in the cell, the main pathways that exist are that DNA can replicate itself, so that's called semi-conservative replication, and also DNA can be copied into RNA, that's called transcription, and RNA can be used to make protein, that's translation. In fact, transcription is the only way that RNA can be made, and translation is the only way that protein can be made, and replication is the only way that DNA can be made, except there is a reverse transcription. But anyway, but these are the major pathways. The special pathways that are pretty rare, reverse transcription is something you see in retrovirus that can copy an RNA molecule into a DNA copy. So that's pretty rare. There is an RNA replication process, and then of course RNA can be used to make protein, but these are pretty specific and special to only a very limited number of organisms.